Hello everyone, I'm Anas Al-Ubaidi, and the title or the topic for this lecture or session is Protocol Development and Application Advanced Review. And this lecture or this session is within the chapter that is about the introduction to clinical research and regulatory frameworks. And of course, this lecture and the chapter is part of the Advanced ICH Good Clinical Practice Certification course program by CCRPS. Introduction. The protocol is the foundational document in clinical trials. It provides the study protocol or the trial protocol provides detailed guidance on how the trial will be conducted and ensures that the study follows regulatory, ethical, and scientific standards. In this section, we will delve into the key components of protocol development, its importance, and its practical applications in clinical research. Key points. First key point to start with, key components of a clinical trial protocol. Title and identification, the purpose. Clear title and protocol number to identify the trial. Example, a protocol for a study on a new diabetes drug would be titled Randomized Controlled Trial of XYZ for type 2 diabetes. Just for an example. Just as an example. Background and rationale. Purpose describes the medical or scientific need for the trial, supporting data and hypothesis. Example, the background might include data showing that current treatments for type 2 diabetes are insufficient in controlling blood sugar levels in certain populations. Objectives, the purpose here clearly states the primary and secondary objectives. Example, a primary objective could be to determine the effectiveness of XYZ drug, which is the investigational product or the investigational drug or the study medication, SM. IP or SM, the same, and lowering blood sugar levels, and the secondary objective could be to assess the drug safety profile. And here is a diagram on clinical trial components. Study design. The purpose describes the trial design, including randomization, blinding, and control groups. Example, a double-blind placebo-controlled study design is commonly used to eliminate bias in treatment assignment. Eligibility criteria. Purpose defines who can participate in the study. Inclusion and exclusion criteria. Example, patients aged 40 to 70 with diagnosed type 2 diabetes, excluding those with severe renal impairment. Interventions. Purpose specifies the treatment or intervention to be studied. Example, the experimental treatment is XYZ drug, which will be administered orally at a dosage of 50 milligram daily. As an example. Outcome measures. Purpose defines how the primary and secondary outcomes will be measured. Example, primary outcome could be the reduction in HbA1c levels at 12 weeks, and secondary outcomes could include adverse events or changes in fasting blood glucose. Statistical analysis plan. Purpose here describes how data will be analyzed, including statistical tests and significance thresholds. Example, a t-test could be used to compare the means of HbA1c levels between the treatment and control groups. Ethical considerations. Purpose here ensures patient safety, informed consent, and ethical review by an independent ethics committee like IRB, Institutional Review Board. Example, participants will provide written informed consent after receiving full details of the study and its potential risks. Study management and monitoring. Purpose details how the study will be monitored to ensure compliance with the protocol. Example, regular monitoring visits by a clinical research associate, or a CRA, short for clinical research associate, which will be conducting the CRA. The CRA, the clinical research clinical research associate, CRA, clinical research associate, will be regularly conducting monitoring visits. Could be both in person and virtual, mostly in person. Usually, maybe all of them in person, but nowadays could be a combination or a blend or hybrid, uh, both in person, physically in person, and virtual, remote. So regular monitoring visits by a clinical research associate, CRA, to ensure adherence to the protocol. And here's a diagram on components of a research study. Application of protocol in clinical trials. Protocol as a blueprint for conducting research. The protocol acts as a guide for all activities in the clinical trial, from patient recruitment to data analysis. Example, if a protocol specifies that only patients with HbA1c levels above 7% are eligible, the research team must ensure that all enrolled participants, subjects meet this criterion before any treatment is administered. Protocol deviations and amendments. Deviations occur when actions taken during the trial do not strictly follow the protocol, such as administering a drug at the wrong time. Amendments are informal changes to the protocol made after the trial has started. Example, if an unforeseen side effect emerges, the study team may amend the protocol to limit patient eligibility or adjust monitoring requirements. Protocol compliance and monitoring. Regular monitoring is essential to ensure that the protocol is being followed, and usually, again, as we said in the previous slide, by CRA, clinical research associate, CRA. Example, if a participant's medical condition changes during the study, the investigator must assess whether it affects protocol adherence and make necessary adjustments. Common protocol challenges here: recruitment issues, inability to recruit enough eligible participants within the prescribed timeline. Example, a protocol specifying that only patients aged 50 to 65 with a specific medical condition may be enrolled may limit recruitment due to the narrow age range. Protocol complexity. Overly complex protocols can increase the risk of deviations. Example, if the eligibility criteria are too stringent or the intervention requires complex administration, compliance may be difficult for investigators. Summary table or tables. Here we have a table on key components of a protocol, a table on protocol deviation examples, table on protocol amendment examples, a table on application of protocol and study conduct, a table on common protocol challenges and solutions, and then we have the MCQs, multiple choice questions, in order to revise what we have covered through this lecture. Question 1. Which of the following is not typically a component of a clinical trial protocol? Title and identification, definitely it's there. Statistical analysis plan, definitely it's there. Eligibility criteria, yes. Inclusion and exclusion criteria. So the answer is C, clinical trial report. A clinical trial report is separate from the protocol. The protocol includes trial design, objectives, and other study details. Question 2. What is the primary purpose of a protocol amendment in clinical trials? Would be B, to correct an error in the initial study design. So B is the answer, as amendments are typically made to correct design flaws or adapt to unforeseen circumstances while maintaining ethical standards. Question 3. Which of the following best describes an example of protocol deviation? Would be B, enrolling a participant or a subject who doesn't meet the eligibility criteria. Enrolling a participant who doesn't meet the eligibility criteria, the inclusion and exclusion criteria, is a protocol deviation as it violates the study's guidelines. Question 4. Which of the following is a common protocol amendment? Would be C, for instance. C is the most appropriate answer here. Expanding the age range of eligible participants is the most common protocol amendment. C is the answer, as expanding the age range of participants is a valid amendment to increase recruitment while maintaining the scientific integrity of the study. Question 5, what is the primary benefit of following the clinical trial protocol closely? Would be B, ensuring the trial's ethical compliance and data integrity, as adhering strictly to the protocol ensures that the trial is conducted ethically and the data collected is reliable for analysis. And here we come to end of this lecture. Thank you very much and goodbye.